Okay, in the first phase of glycolysis, glucose enters the cytosol and gets catalyzed by hexokinase. And in this reaction, ATP gets turned into ADP and turns into glucose 6-phosphate. Then in the next reaction, it gets converted by isomerase, but this is just a simple rearrangement reaction, so no energy is used. And so it turns into fructose 6-phosphate. Then it's catalyzed by phosphofructokinase, and again, uses energy, so it turns ATP into ADP, and gets turned into fructose 1,6-biphosphate. Then it is catalyzed by aldolase and gets split into two molecules, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. But eventually, all of the dihydroxyacetone phosphate will get turned into G3P, and these two can be converted into each other using isomerase.
All right, in phase 2, we're going to start with the glyceraldehyde 3-phosphate, which is going to be converted by the GL3P dehydrogenase, where an NAD is going to convert to an NADH where a phosphate group is added. This turns into 1,3-biofosphoglycerate and is converted by the phosphoglycerate kinase, where an ADP converts into an ATP where energy is produced. Next, it turns into a 3-phosphoglycerate, which is then converted by the 3-phosphoglycerate mutase. This is just a simple rearrangement reaction, so you could think of mutase as sort of like a mutation, that it turns into a 2-phosphoglycerate. Next, that is then converted by enolase and magnesium, which will turn into a phosphophenylpyruvate. Last, the phosphoenolpyruvate is converted by the pyruvate kinase, where ADP turns into ATP, where another energy is produced. Finally, you'll end up with a pyruvate, and then this cycle will repeat itself to make 2 pyruvates.